A very good afternoon, one and all. My warmest welcome to each and everyone present here to the seventh day of Golden Jubilee Lecture Series conducted by the Department of Seed Science and Technology, College of Agriculture, Vellanikara. First and foremost, I invite Dr. D.G. Bastien, Professor and Head, Department of Seed Science and Technology, College of Agriculture, Vellanikara, to deliver the welcome address. Welcome, ma'am.
Thank you, Vaishnavi. Good afternoon, and I welcome everyone to the latest edition of the webinar series. At the outset, I would like to welcome our chief guest speaker of the day, Dr. Yasin Jessima K, who is a very good friend to me for the past 2 decades. Next, I would like to invite Dr. Rosemary Francis, Professor, Agriculture Research Station, Manuti, who is actually the brain behind this webinar. And she was the one who took the lead to collect all these. Everything behind this webinar is credit goes to Dr. Rosemary Francis. So I extend a hearty welcome to you, ma'am. I welcome Ms. Fahida PK, the Assistant Professor at the Department of Seed Science and Technology, and Ms. Agina Goban, who will be introducing our Honorable Speaker of the Day to this webinar series. Last but never the least, I welcome all the participants to this webinar and hope we will have fruitful discussions today as we have had in all the previous sessions. So over to Ms. Agina Gopan.
Thank you, DG ma'am. A very good afternoon to all of you and a warm welcome to our honorable speaker. So it's a moment of immense pleasure for me to introduce our speaker of the day, Dr. Yasin Jessima K, Scientist SS, Division of Genomic Research, ICR, NBPGR, New Delhi. She is a native of Madurai and her post-graduation and graduation was from Madurai campus, Tamil Nadu Agriculture University. And it is also, we are lucky that she is also an alumni of our university, as she did her doctoral program from Vellaini campus, Kerala Agriculture University. And she took her postdoctoral fellowship from ARO, Ministry of Agriculture, Israel. She is a recipient of JNU JRF, ICR SRF, ARS, and International Postdoctoral Fellowship. She is an excellent and passionate researcher with international recognition, and she has published more than 50 research papers, and she is an author of more than 5 books. And for incredible excellence in this area, she was honored with more than 22 awards. Her research areas are development of genomic tools, databases, and bioinformatics for crop improvement in legume crops. It was Dr. Wasin ma'am and her co-workers who first introduced the concept on smart agriculture for nutritional security in 2013. Ma'am, we are very grateful that you accepted our invitations for this online lecture series. So I once again, on behalf of Kerala Agriculture University, invite you for this wonderful lecture series. Thank you, ma'am.
Do I have the permissions to start now?
Yes, Yasin, please.
Please let me know if you can see my slides.
It's visible.
Very good afternoon to all of you, respected head studies, professors, and my dear friends. I'm highly excited to share my research experience in this area of genes associated with dormancy and germination. Being a person who is sitting on the National Gene Bank of India, we do have the privilege of working on seeds because we depend on seeds. We store seeds, we process seeds, and we share seeds. So we are the people who are working on seeds, and we do the same thing to analyze the molecular profiling, the variations, and other things using a seed material. starting from DNA fingerprinting and our genomic resource. Everything depends on seeds. Not just as a researcher, being as a human being also, what we consume is almost more than 80% seeds, whether it is processed or unprocessed or killed or a fresh live seed. We are consuming it daily. So the world depends on seed, and why not we being a scientist? In this presentation, I'll be using some of the terminologies most of you might be familiar as because you're all in the research area. But for the freshers, younger students, I'm just explaining it again. We know the terms ecodormancy, endodormancy, and other things. This ecodormancy is something related to the environmental condition which is imposing some stress on the living material. And this ecodormancy includes everything like a stress condition, a drought condition, or a winter, or temperature stress, whatever it is. If you say like we are working on moisture stress, especially my lab concentrates on both the deficit moisture stress as well as surplus moisture stress. So we take this ecodormancy. This is where the starting point we started working on this kind of research. So how this dormancy is exactly related to the field-related condition, you may ask. The thing is, a dormancy is something which is a live material in a quiescent state. Live material which is restricting its biological activities to a quiescent state. And it is nearing to something like a vitrification stage of the in vitro callus condition. So the people who are working on tissue culture might be knowing very clearly, like once the callus reaches its vitrified stage, at that stage, then getting a regeneration is a difficult task. Like you have to again convert it to certain stages. That means it is a sleepy stage. So everything is similar. The mechanism is similar, whether it is a seed dormancy or a bud dormancy or something which is faced by the plant during its stress condition. So almost mechanism is similar. And we all know that during bud dormancy, it is not just dormancy. The plant is getting prepared to produce flowers. That means like there is floral parts getting developed inside the bud. So that is a kind of synthesis stage. So that kind of similar mechanism is experienced under stress condition also. This is our finding. That's how we ended up with some genes picked for the germination and dormancy related things. And next one is delayed dormancy. This is a little bit unwanted thing just because when we sow the seeds in the field, we expect everything to germinate immediately. And one more thing which is unwanted is the fresh seed dormancy. Means actually we need fresh seed dormancy, but if it is germinating in the field, then we may not be having anything on our plate to eat. So that is needed. Like fresh seed should have the dormancy, whereas the delayed dormancy should not be there. So we need both these things. And one more thing is precocious germination. The best example what we can say is in rice field and wheat field, if it is raining during harvesting period, you can see the sprouts in the field itself. In the panicle itself, you can see the sprouts. Just like that, what you have seen in what you can see in Zyzium mediole. Okay, so in that crop, the fruit itself will be having a plantlet. So just like Avicennia, rhizophora, all those things will be having the plantlets. Okay, so just like that, it is also having. So that is not needed in our crops because we cannot eat seedlings. We need grains to eat. And the next one is imbibition. Imbibition is not exactly the germination process. It is just only taking water. Okay, so it is a stage prior to the germination. And next term is association, whole genome sequencing, and SNPs. Association is something we are having the phenotype as well as the genotype. Whether this phenotype and genotype may be of different terms, whether it is a biochemical character or a phenotypic trait, what we have observed in the field, or in case if it is a SNPs taken from the genome or something else is also there. I will explain it later. The next is germination. Germination is a three-way process. Like there are different phases of it. We will see in detail. The next is gene expression. Gene expression, in majority of the cases, it was studied at mRNA level. That means like we do have mechanisms or techniques to quantify the level of expression in mRNA. And then we say that it is expressed. But the thing is, actually, having an mRNA is not a confirmation, because we do have, there are so many role plays in that, like non-coding RNAs, sponges, and there are other proteins which pack them and carry them away from the cell, like it is being transported somewhere else. So all these things are happening. So gene expression is not exactly directly the proteins present in the cell. That means like what is present in the mRNA is not exactly will be the protein. Okay, the protein can be of totally different. Means you will be having the mRNA, not the protein. So that kind of situations can happen. Then almost we are going to discuss about gene expression during germination. Mostly there are some phase in between. The thing is like during maturation and after ripening, there is one stage after ripening. That is after maturation, the period up to its germination is known as after ripening. And this after ripening facilitates to have some sort of dormancy for a period so that we can store it, we can consume it. All these things can happen during this after ripening period. And we can use it as a seed for the next season. Because once the season is over, immediately after that, if the seed germinates, then the condition may not be favorable for the plant, and that is an unwanted condition. Then expression, transcriptome. Transcriptome is the total mRNA. Okay, so we check mostly the transcriptome means mRNA. Otherwise, nowadays we study RNA-seq. That includes all non-coding RNAs. The junks, what we thought so far is not at all a junk, and all those things are present in the RNA-seq, whereas the transcriptome is only the mRNA. And ripening, I said, like after maturation, there are some process which packs the mRNA also, and there is no just further production of this. Ribosomes won't be active, whereas the polysomes will be saved. Okay, and these things as well as the proteins what is stored during the maturation stage, and these things help in facilitating the germination. So all these things happens. After ripening, I said like this is going to help you in storing. So if you want to contact me regarding any of the research or anything else, then you can catch me there in the ResearchGate or from my institute's webpage. And as you know, like I do a lot of Googling. So I said like we started working with the seeds. That's how we ended up with some of the genes picked for dormancy related things. So some of our previous publications started with a very basic thing like long-term storage, how it is differing, all those things we started, and how to break dormancy. So, okay, how we can store it and what kind of procedures will help you in breaking the dormancy. So once we started doing all these lab work, then we started understanding slowly, like there is something special inside the seed that we have to decipher. And once at the simultaneous time when we were working on this abiotic stress conditions, we happened to find out some of the genes getting expressed in the same time, like something similar happened. Similar mechanism we have cracked. And we have published also some of the things we have already published, or something we kept it in the bag, and so that you can expect sooner some of the fascinating or exciting results from our lab. I'll just, so you got to know that I'm a scientist, I'm here in NBPGR, but some of the publications I just wanted to show like what kind of research work I'm doing so that you can understand how I am identifying some of the genes from these kind of things. So one such recent publication is we have mapped the long non-coding RNAs as well as link RNAs, and then we have identified microRNAs. And this is our recent publication. This is in PGNP. Similar work we have done in macrotyloma also. Here we have reported, this is our first report of the set of microRNAs which is actually contributing for differential stress response. This work actually took a long time, because we started with a total gene bank collection. From that, we have developed a core, and from that core, we have identified some contrast, and those contrasts were for the moisture stress response. Like identifying a susceptible germplasm for moisture stress was a difficult task, just because the nature of this crop itself is a drought tolerant one. And from that, we picked somewhere around 20, and then we have grown them in the growth chamber. Then I isolated RNA. We did RNA-seq analysis, and then we picked some of the genes out of it, and then we have cracked the mechanism. Like this is actually happens, and then that has been validated by real-time expression, as well as we have done some of the cytological work also to prove that this is correlating with this. So this is the actual mechanism. So that kind of work we have done in horse gram. And apart from that, we work on developing new lines, like somewhere around 65 high-yielding lines we have developed in PGNP. And almost like we being scientists from NBPG, we cannot release it directly as a variety. But we have developed some of these lines. Almost we will be nearing to registration of those lines, so that at least this will be available for the breeders. So that we can do. And we have identified all those things, and we have developed somewhere around 7 lakh plus SNP markers. That markers are available in European Variants Archive. That complete map we have published in Variants Archive. It is openly available. And using that, our partners filtered markers, and then they developed some of the chips, and then we have validated that with our high-yielding lines also. So that is also present in this. This is one of our high-yielding line under rain-fed condition. I cannot say completely a drought condition, because there was no rain out shelter. Almost, it might have faced some of rains also. So it's a high-yielding line, and its plant structure is so small. And even it is grown in very fertile condition. It will be somewhere around 60 to 75 centimeter, hardly, not more than a meter. So that's how it is. And this is a single season crop. Like PGNP, if you are allowing, or if it is grown in different conditions, it will grow like a tree. But this plant is a determinate type. And I said, like, we have developed some 7 lakh kind of SNPs, and those SNPs are present in European Variants Archive that you can see. And similar work we have done in some other crops also. And we have developed some browsers. This is available in LIS, that is in USDA's webpage. And we help our collaborators in doing some of the genome assembly and other kind of things also. Some books we have published, book chapters. And almost everywhere, what I start is from gene bank collection, and I will end up in a system biology approach. Like nowhere it is a single unit, like not starting from 1 to 1. We start from a bulk, like thousands of accessions, and then we end up with a few hundreds or tens of genes. And from that, we pick single ones. Like just like I have mentioned, 65 high yielding lines or 20 stress responsive contrast from Hasdam. So just like that, we end up with a few genes. Like these genes are highly amenable. Like this can be used in modifying the crop, or you can design a crop, you can edit a crop, you can edit a genome. So that's how we end up with our conclusion. And this is one such thing what I have done in Arrakis. And this work actually I did for my colleague, and this paper is not yet published. I think they are taking some more time to do that. Here we have taken some of the feeding experiments. Like we have taken 2 different species of Cajanus, and then we have studied what will be the impact of pest attack, and that kind of expression analysis we did. And just like that, we did differential expression analysis in some of the legumes also. Like mostly my work concentrate on legumes, so you can see that. And earlier we have reviewed one work, how to edit genome, like what are the possibilities, how to do that. and mostly we work on markers and maps and something like that. Something, if it is exciting research, then we will write even e-letters. We do publish databases, and with lot of markers, lakhs and lakhs of markers. So far we have crossed more than 10 lakh markers, and all those markers are already publicly available in public domain. It is openly available. You can go through that once you need all these markers. If you cannot find out, you can directly contact me. Then this is another example. Okay, so I said, like, we start with a bulk and then we end up with a tens. At last we ended up with only one protein, and this protein was found to confer something related to abiotic stress and something related to quiescent state. And the exact mechanism is not yet cracked. Sooner I think I'll be able to finish it this. So one more publication is expected in this area. And yeah, this is another marker related work. At last, again, what I said, to the same point I reached, like how we started with the total gene bank collection and how we filtered. Mostly I go for geostratification to pick a set of accessions. So from pigeon pea growing all 16,000, even 16,000 may not be available. So something like if we ask for the same seed material, somewhere around 12,000 may be available or 13,000 may be available. The others may be in a multiplication state or different stages, so that they cannot share it. So what we do is I start with the geostratification, and then I started taking somewhere around 2,000 accessions. Of these 2,000 accessions, something were from my lab, some breeding material. So all those things we accumulated together and then grown in multiple locations, somewhere around 6 agroclimatic zones of India. We have grown all these things, and then we have identified. So it was a grueling struggle kind of thing. Then at last we ended up with a handful of diverse best performing lines, and most of these lines were from our lab. And you see, like, we are taking a hot sun bath during summer, and with all our beautiful dwarf high yielding pigeon pea lines. So this looks like a soybean plant actually, but this is a pigeon pea line. Like normally pigeon pea will be like a tree, but we have developed some of these dwarf lines. And we did some of the crosses, and then we have developed different high yielding lines. And so this plant is the same one what you have seen in the previous images. This is a high yielding plant at maturity. I'm standing there and making some observation. And we work with extreme conditions. One is drought condition, that is deficit moisture stress, and another one is flooding condition. So we have developed a line for flood tolerance also. So this was tested for almost like normally flood tolerance means in legumes not more than 2 or 3 days, but this crop survived more than a month standing water. And the mechanism is also cracked, like how it is withstanding that much flooding tolerance. So we have developed some of the high yielding lines. I have shown here how it is performing over the checks. You can see the checks at the right side end. And as I said earlier, like we have done some SNP analysis with our collaborators, and all these groups are having our lines. You can find out DGRG lines. All those DGRG lines are from our lab. And we ended up with networks, and from these networks we identify the nodes. That means like the effective genes which can help in designing a new plant. So that's how we reach. And what we have identified is exactly related to dormancy. So again, back to the point, like topic of today's discussion. So seed is the starting point of everything. The globe is surviving just because of seed. Everybody is surviving just because of seed. So without seed, we cannot imagine this green globe. Okay. So whether it is green or yet to be made into green, that is in our hand. And we are everywhere talking about nutritional security. And as the introducer informed here, like we started coining the term smart agriculture for nutritional security, and that paper is in current science, you can go through that. And I place one request also before getting into the topic. Please don't take it another way. Like before discussing the subject, we are putting some request. So like plant all fruit trees in your campus instead of just shade providing trees, so that at least people can have some of these fruits. Like no need to just break the chain between food and money. So that is very much essential to have nutritional security. And those who are interested can see some of my lectures available in YouTube related to that topic. And the dormancy is actually a evolutionary significant event, like that is needed to a certain limit, and that is not needed to beyond a limit. So that is the important point. So we will take one more example to discuss that also. And as I said earlier, germination is a multi-phase activity. In the first phase, actually, it takes rapidly the water. That means imbibition happens. In the second phase, actually, the first phase, what exactly is present in the cell is there will be a lot of Lea proteins and some heat shock proteins will be there. Small heat shock proteins will be there, and some oxidoreductases will be active. So only that much will be there in the first phase. Means there is no germination exactly, other than taking water inside. And in the second phase, there will be DNA repair and transcription will be initiated, translation will be initiated, and there will be DNA synthesis will be started, and the stored food material, what are the stored material available inside the cell, all these things will get starting its, means it will start its metabolic process, and there will be expansion of cell size. So everything happens. That means it is taking a living condition, like from quiescent state to active state, it is changing in the phase 2. Whereas in the phase 3, the actual germination process happens, like the embryonic axis, all those things will start growing, so that you will have a viable seedling at the end of phase 3. So this is actually the original germination what we talk about. So many things related to germination will directly fall into 3 hormones: auxins, AB, as well as GA. And these hormones play a major role in making the seed, whether it is dormant or whether to repress its germination or to help them fasten their germination. So the ratio between GA and ABA, GA by ABA is actually playing the role, and there are no clear-cut pathway for this. You can see a lot of dotted lines. All these dotted lines means there is discontinuous chain. Okay, so something is missing in that area, and they have to find out where it is, what it is. So that kind of thing is there. And almost there are 2 things, zeaxanthin as well as one zinc finger. So only these 2 genes everybody is pointing out, because they know only that. So what is already reported, that is being researched. It is not researched. So that is researched and almost found again and again. So as I said earlier, like association means not just marker to phenotype. You can associate with anything, like a phenome can be associated with the genome and epigenome, transcriptome, proteome, metabolome. So whatever you are having, everything can be associated. So only the trick is data analysis. And so at last, what you are having as SNP means, then this can be directly associated to a particular phenotype, particular trait. But the actual thing is how you are interpreting your result. So if you are having a QTL, the QTL can be of very big or long region. And in this region, somewhere we can say something is present and that is contributing. That's all. Like you cannot say like what is present and what is contributing. Whereas if you go for an association mapping, you can clearly say what is present and how it is actually contributing. So that much you can say. So association mapping is a preferred technique over QTL, and that should be supported. Means we have to prefer association mapping over QTL analysis. So once you arrive at Manhattan plot, then you will be having your SNPs located, and what is present above zero point 0.5 is all significant, and you can trace back these SNPs to the particular region, and from this region you can find out to pinpoint your exact genomic loci or the exact gene of interest that you can find out. And then you say like this is the one what is actually contributing. But the trick is how you are picking your loci from the Manhattan plot. That is the thing, crucial thing. And if it is not a single one, then you can go for system biology approach. From this, you can go for expression analysis, or from expression analysis, you can come for system biology approach. In both way, you can do. And at last, you can find out a node, and that node can be edited and edit to find out whether it is actually contributing or not. So that kind of example also like what we have done with one of our work. So this is the best example. This is Arachis sublobeta, something like that. Okay. So they have taken two different species, two different contrasts of this species. And one is dormant one, that is fresh seed dormancy. Okay. Like it is not germinating in the field itself. So one is having fresh seed dormancy, whereas the other one is having lack of that fresh seed dormancy. That means it will germinate in the field itself. So there are two contrasts they have taken, and these two contrasts were crossed. They made a RIL and they identified a QTL, and apart from that, they did again whole genome sequencing. They have identified SNPs and they have associated that trait. And at last, what they did is they linked that to the QTL. Means using SNP analysis and whole genome analysis, they have not found something new. What they have identified in the QTL itself, they have validated. Yes, this QTL, this region is contributing. So what they did is exactly they have plotted everything. They made they have identified some thousands of SNPs, and then they said like this region is contributing. This is B02 pseudomolecule, I think. And this region is contributing. Why only this? If they are taking it based on this 0.5, they might have taken all these things and they did something system biology approach. They didn't. But in our analysis, what we got is something from this area, like in the blue mark what I have given. Okay, so this is what we got, not this one. This is not correlating. This one is correlating with the previous reports, not with actual trait. Okay, so this is also having association. This is also getting co-expressed, but this may not be the one which is actually contributing for the trait. So this is our finding. So you can expect something. It will take some more years. Yes, I'll do it after 2-3 years only. So it will take some more years. So after 2-3 years, if you read, then you can get some fascinating results from my lab. So something like that they have identified, and in one more molecule also they did the same thing, but whereas they have not even shown the Manhattan plot so that I can spot it. That they have not shown directly. They have plotted the gene, and then they have done some expression analysis. It's okay, fine. According to their results, their purpose is to prove that their QTL is having something which is contributing for the trait. So yes, that is doing that. The purpose is solved. And this is another example. This is a sleepy mutant. I think it is a LER background in Arabidopsis. All these things are transcriptome analysis. One thing is like everything is compared with the contrast. The first one is a double mutant with wild type, and this is with the dormant one, and this is with after ripening and dormant, and after ripening with another dormant. So these three things are different. Okay, so this is a sleepy mutant, whereas a wild they have compared. And this sleepy is a box protein kind of thing. Okay, so this box protein, actually what it does is it is working something like GA insensitive. So GA insensitive means obviously this will affect your GA-ABA ratio, and just by that it can repress germination. Okay, so that's what actually happening. And I said like box protein. Okay, we have worked with some of these F box proteins. This is from my collaborators lab. Um, Actually, there was some very interesting mutants, and these mutants had variations in this SD1 as well as F box protein region. And then we have picked something, and that was done with the genome editing at last. Then function is confirmed, like it is contributing for dwarfness, as well as that is getting expressed in the seed also. So something related to that sleepy mutant, something is exciting. But this F box is not exactly characterized, in the sense like we have not given exactly like some names or something like that. So it's a very, very good loci what we have identified. Then there are so many other reports, like they report lot of thousands of hundreds of upregulated expressed genes and downregulated genes here and all. Like what are all the things in blue, it's downregulated, and what is in pink, it is all upregulated. So you can see like during after ripening and dormancy and during germination, what kind of things are happening. Almost many of these things are like we cannot exactly say like false positives, but this will be upregulated. This is a co-expressed one, co-localized one. And all these things are exactly not contributing for the trait, but under the same condition, it will be expressed. It may be doing its own function, but not contributing for the trait. So there is a need for some other specific approach to pick exactly the gene of interest. So here you can see like myosin. Myosin is like during all active stage, you can see how it is, and almost here it is differentially expressed. So just because it is in vitrified or casein state, so obviously it will be there. And this kind of result is similar to exactly what we have identified in our abiotic stress conditions. Okay, like what exactly is present is something is overall silent. Something is overall silent, means it is keeping quiet. It is shutting down all its additional burden. So that kind of situation is happening exactly in all these conditions, like whether it is a stress condition or a germination or a dormant condition or a bud dormancy or flower development. So that you can see like some genes we have reported already that is showing exactly similar to the dormancy. And there are some reports where they have illustrated some of the mechanism, saying that there is evidence that this gene is contributing. Obviously, in a pathway, if you cut anywhere, obviously there will be an effect. Okay, so like we cannot say exactly like that point is only that point is leading to the end point. Like there are two starting points and end points you do have, like in a transport or some roads you are having. Okay, so anywhere you cut, definitely the road will be broken. Okay, so anywhere if it is a break, the road will be broken, and you cannot say that only that is leading to the final stage. It is not like that. So something different is actually happening, and this gene is actually contributing, but we cannot say like only this is contributing, because everybody is working with this zeaxanthin epoxidase, and almost everything will be reported, like dormancy related, germination related, auxin, yeah, all hormones, everything. If you see, like there will be upregulation, downregulation, but the thing is, the magic is how to filter them to find out the exact key role player. Okay, so far in the discussion, what we have seen is like people have identified some of the contrasts. They have identified some upregulated genes, downregulated genes. They used some knockout mechanism to find out how it is working. and then transgenic approaches also. So all these things they have done. But in the newer generation of research approaches, everything is related to something epigenetic also. So there are some methylation dependent and other kind of epigenetic actions which is actually happening during this dormancy, which is being relieved during its germination. So that kind of association is also there. And from our reports, from our results, what we have identified is there are non-coding RNAs which is actually contributing for this mechanism. So something what we have reported in our Hasdam work is also reported in the dormancy. So the recent report, some papers we could see, like out of 15 microRNAs what we have deciphered from Hasdam, 1 or 2 were directly identified in the dormancy condition. So that we could able to validate in our lab also. So that also we have done. This is another example. They did exogenous supply of auxin, and then they did some identification of genes. All these genes are something related to ABA biosynthesis pathway. And using this, like what they did is they supplied IAA and they tested the seeds with two repressor molecules. One is floridone. Floridone is a ABA repressor, whereas baclobutrasol they used that baclobutrasol is a GA repressor. So these two repressors they used, then they found the fluctuation like GA by ABA fluctuation, and just by that they found that decline in GA and increased ABA represses germination just by addition of exogenous auxin. So this is their finding. So that they have done means exactly you can see in the pictures also, like in the presence of contrast as well as inhibitors, how it is exactly functioning in the germination condition. So just like that, what I said in the earlier condition also, during bud dormancy, from outside it is dormant, but inside the floral parts are getting developed. Mostly the pollen and other parts are getting developed. Means androecium is getting evolved or developed inside during this bud dormancy period. And the differential expression reported somewhere around 1000, okay, somewhere around 600 to 1000 differentially expressed genes, and almost 99 of those genes were found associated with CR loci, and this CR loci is winter hardiness loci, and DAM 4, 5, 6 are the major genes they have reported, and these genes are like again the F box like proteins, okay, something like that. And in many cases people talk about DELA proteins also. So there are key role players, and it is yet to be deciphered. It is an infant stage, means as far as my experience is concerned, this is in infancy. So there is lot of expectation in the future from these kind of research, so that something can be found. Means like if we are having some very good genes and mechanisms which can be either exogenously or editing wise, whether it can be altered, then that can help in conservation also in future. So that kind of things has been deciphered. Means the need for the future research is deciphered, not exactly the key role players. And this is from my finding. I'm not disclosing all my results. I'm just showing only this network. And this is a gene, what I have identified, this one as well as this one. So these two we have identified as flowering regulators, and these flowering regulators are actually contributing for dormancy also. And just I said like epigenetic events actually regulate dormancy, and some basic research has been done. Means not, I cannot say basic research, some research has been done in this area to prove that that is contributing exactly for dormancy. you can see that the methylation pattern and where it is actually happening. Okay, so what we have identified is some markers. I said, like by system biology approach, we have identified some of the nodes, and these nodes, if it is edited, can contribute for breaking the dormancy or enhancing the dormancy. And this has some splice variants also. Splice variants you might be knowing, once the initial mRNA is coming, after that there will be action of spliceosomes, and these spliceosomes cut in alternative way during different conditions. There can be a spatio-temporal regulation for this, and just by these regulation you can get alternative splice variants, and these splice variants will contribute for different proteins. Like you will be having different splice variants producing different proteins in different conditions, so that this function of these protein will be exhibited in the different way. So this is also another finding what we have identified. Apart from that, the key role players are the microRNAs and other non-coding RNAs as sponges, which is present. Here, the primary microRNA comes and it forms duplex breaks, and then like double strand small RNAs are present in plants, and this is getting exported to the cytoplasm using exportin, and then from there it starts clipping the mRNA, so that prevents function of a particular mRNA and prevents its translation, so that a particular function is prevented, so that a different phenotype is expressed. So this result, we are yet to finish this research, so it will take some more time for us to publish this work. Once it is published, you can read it from my personal pages. Thank you. If you are having questions, we can discuss now.
Thank you, Yasin. Hello.
Yes, ma'am.
Thank you very much. Now I request the audience to click on the raise your hand button, so that you can interact with the speaker, or you may post your queries in the chat box, and she will answer it for you. Am I audible?
Yes, ma'am, you are audible.
Digi, can I ask a question?
Yes, please. Yes, please.
Yeah, Yasin, hello.
Yes, ma'am.
Yeah, what thing is this differential expression? We are trying to decipher the genes associated with all these dormancy and germination. What would you think? How would it help the seed technologist? Like, because this is a seed technology group. In what way do you think this would help a seed technologist?
In case if you are having a major problem in conserving some sort of seeds, then you can pick some contrast to find out what is exactly is present, and then you can suggest further for breeding work, like this is a donor for a favorable trait, so that further breeding work can be done using these things, so that conservation and future use will be of ease and good for mankind. So that kind of decision can be made using that.
Yeah. So the recalcitrant seeds, they never seem to sleep, right? So they keep germinating. There's a lack of dormancy there. So in that line, do you think this can help, and in what way is it being worked upon?
We can bring them to quiescent state by exogenous supply of hormones. So that is possible. And just like slow growth cultures, we can maintain them in vitrified stage.
So can there be a manipulating? Can we manipulate them genetically?
We can manipulate them, and this is related to your previous question also. Like if some mechanism is identified for how we can manipulate, or what is exactly playing the key role, then we can easily manipulate it.
Yeah.
So no need to have a genome edited plant. Like that's why I have taken one example of exogenous supply of oxygen.
Exogenous. Yes, yes. So is that being worked out, or have they tried this in NPPGA, because you are dealing with a lot of
No.
different?
That one is a published example.
Acha, acha. So no work of that sort in NPPGA?
Uh, that we have to ask conservation people, because I don't think they are working, because they work only on long term conservation. Like I have shown some of the example that is with my collaborators from conservation division. Like somewhere around 16 years 18 years conserved material we have taken, and then we have seen how far it is intact, or whether it is stable, something like that. So only that kind of research is happening at present. Maybe they may be doing it in future.
Okay, thank you. I think there are many others waiting to ask you. So let me drop.
Okay. Drying of seed after harvest will induce dormancy in rice varieties. Yes, obviously, because the moisture level plays a major role in maintaining it in quiescent state. That's why I said I correlated that with a abiotic stress condition. Like once it is in abiotic stress condition, it is resembling to the quiescent state what is present in seed dormancy. So mechanism is similar. Obviously, drying will induce dormancy. I think whether I answered your question, is that sufficient enough?
Ambli, you can interact, Ambli.
Okay, welcome.
Dr. Yasin, there is a query in the chat box.
Okay, ma'am. I'll see. Myself, Dr. Suresh. Last lecture, the speaker told that government is trying to have 100% seed germination in all hybrids and varieties selling by private seed companies, and that is going to be under law. Can I have your view? Seed germination, that is
One second, Dr. Yasin. I think Dr. Shankaran has raised a hand. We let him interact and then you can come back to Dr.
okay, ma'am.
Shailesh.
Okay, sure, ma'am.
Shankaran, sir, please. Shankaran, sir. Okay, you can answer Dr. Shailesh query, Dr. Yasin.
Actually, dormancy is something different, whereas germination is different. And as much as we are concerned, the minimum requirement is 95% germination. And the private companies are charging very high rate, and they should ensure 100%, almost 99.9% they should ensure. So that is important, I think. There cannot be any difference opinion in that. Even in gene bank, after conservation of so many years, people test it whether it is having a required amount of germination. So that is needed. Yeah.
Actually, the previous lecture, Sundareswaran sir, that is the director of seeds, at TNAU. Yeah, he was giving us a lecture on the research aspects that we could work on in seed.
Okay.
So that is when he was telling that because we are charging people for, we are paying, farmers are paying for the seed. They like when we buy something, we expect it to be in the best condition. So
Yes.
that is where he mentioned that a time will come when people will insist that the seed that you sell them, because he has been paying for it, will have to have 100% germination. So that is what. So that he was prompting us, or rather motivating us to produce seeds that will germinate 100%. So that is why I think Shailesh, Mrs. Shailesh was referring to that, and that's the query he was putting.
Yeah, that is needed. Obviously, it is a valid point. In fact,
Dr. Sailesh, I think you can join in the discussion and tell us how you feel from the private sector.
in what way it will help? It's all genomics work.
No, but still we can always be open to ideas, no?
No. He is asking for PPT slides. So I said,
No, that one we will share at the end of the lecture. Already we are told that at the end we will share all the PPTs and the discussion points also.
And a write-up too, Diji. Yasin, we would like to have a write-up also, because at the end of the compendium of these lectures are being brought out. So
okay, ma'am, I'll send.
Sure, that.
Shankaran sir?
We can ask him to unmute.
Dr. Shankaran sir has pressed the raised hand icon. That's why I thought he must be interested in sharing some points or discussing something with us. Actually, Dr. Yasin, it's a nice topic, but unfortunately, being a Friday afternoon, I think most of the teachers in KU and they are all in undergoing invigilation duty and things like that. That is why the turnout is very less and the interaction is less. That is the problem.
Okay, that's fine.
Normally, it is Saturdays, means nowadays for us Saturday was a holiday, so they are free. And even otherwise, also Saturdays, the lesser number of classes will be there. That is the problem this time we facing.
Here, sometimes on holidays, we face net connectivity problems.
Okay. No problem, no problem. So if there are no more questions or queries, you can write to us if you have any queries later also. I can get Dr. Yasin to answer it for you. And I think now we can move on to the next part of the program. Can we, Dr. Rosemary?
Yes, I am given the duty to sum up what Yazin has been presenting. And to start with, I would thank Yazin for not directly moving into the topic, but rather giving tidbits of wisdom. So the best thing is I liked your phrase about breaking the chain between the food and the, and the money. That's a nice thing that is apart from technology and the thing that we've been discussing later. This is a real good thing that I think it would have stuck with everybody today. So it's a good idea. So I hope it is being practiced in the IRA campus also. Yes, yeah, that's a nice.
No, that is the future plan. Means with that aim only we have written about smart agriculture for nutritional security.
Yeah, yes, I have written down that point, smart agriculture for nutritional security. I'm surely going to Google that and find. You said it is free, right? Yeah, yeah, I will surely. And you said something about YouTube videos also.
Yes,
So,
means
what?
last month I have delivered. I think this month itself. This month, first fortnight of this month, I have delivered one lecture on this topic, and that is available in YouTube. If it is not available, I can get the recorded version also. I can share it with you.
Sure. I think the group will be more interested. I think everybody after this lecture is
There
going to
actually
Google it.
had a lot of participants also, like somewhere around 500, 600 people were there for that lecture.
Okay.
So
So,
that
what
was
was
not
the group
by,
like? What was the
that
occasion
was
for this?
organized by Annamala University.
Haha.
So I can
Okay.
share that lecture.
Right. Thank you. But you said it is on the video. I mean, YouTube, na?
Yes, it is in YouTube. It is
Yeah.
available in YouTube.
And I'm sure after this there will be a heavy rush to look into that YouTube channel. Anyway, it was a nice thing that you started off on a lighter note, and that's most welcome. And then you moved on to, I mean, you have drawn a lot of examples from your own lab, from your own experience and your work. That is really great. And I think it is, you could relate to it more than getting a few examples from outside. So that also was interesting how inherently this epigenetics also plays a role, and the balance between the GA and the ABA when it tips how the germination and the dormancy. There's a relationship, right? It is almost like two sides of a coin. So once the dormancy is down, and then the germination takes over. So there are and as you said, there are a lot of gaps where what really leads to the germination or break of dormancy. There are a lot of dotted lines that people like you, I think more people with like you with more money and access to all the facilities can do better rather than okay, we can have the outputs from your lab. That's more important. And the query that I wanted to, the final thing or outcome is that we need to put that into practical use. So that is why I asked you what about it? How would it be useful? So thank you for that. And also this. You have given us a lot of examples about how the auxin, that role, how it plays in breaking dormancy, and with all of groundnut and PGNP and all those things. So it is really thought provoking. That I think for a starter or somebody who has just who had joined the talk in the middle, things would have gone over their head for me, but then it will slowly sink in. So initially, I remember when people started talking about markers and all those things. No, it never really we would hear about it, but it never used to sink in. But later on, when you keep repeatedly hearing that, it really clicks, right? So this sort of gene expression analysis, miRNA, sponges and things like that, it might have gone over the head for many of them, but surely I think these, when they come refer to this, and then especially when you read articles repeatedly, you are just giving us a small input. And this is only a starter, like when we go to a restaurant, right?
KU I made earlier, and that was on purely on these non-coding RNAs.
Yes. Yeah. So we just start. You have given us an input, and we need to build it ourselves. We built on that. Just like in all classes, we only can give only a very small. You are giving us the tip of the iceberg of information. So we'll consider that as your talk as that, and I think all of us, even including myself, I'm not much aware of. I don't know this topic in detail. So we will work on it, and we will also catch up with you.
Also, because only means from IRAPG school, this course has been started, and only one session we have taken. So
okay.
This course is very new, and only at IRA we have introduced. So I'm handling that course.
Oh, Yeah.
So
You,
this,
we can see how very with ease you are handling this topic, and it is like you're talking to some. It's like a daily affair, no? It
yes, it
makes.
is my
Yeah, that's the ease with which you were talking. So must be. But still, we will catch up with you people later. At least in another 1 or 2 years, we'll also be there with you. Everything will be sinking, and especially there are a lot of juniors in this group. I think they will be undergoing a lot of exposure, trainings, and things, and surely this will really benefit them. Thank you, Yazin. Thank you so much.
Thank you.
Thank you, Daisy, for the opportunity. Thank you.
Thank you, Dr. Rosemary Francis. I welcome Ms. Fahida to express her vote of thanks. Fahida?
Yes, ma'am. No, all. It is a great honor to thank everybody for the successful organization of today's webinar, organized by Department of Seed Science and Technology, College of Agriculture, Kerala Agricultural University. I thank Dr. Yasin Jishima, Scientist, Division of Genomic Resources, NGPGR, New Delhi, for her excellent lecture on the topic Differential Expression of Genes Associated with Dormancy and Germination. Really, the topic was very informative to us. Thanks for giving examples from your actual research findings from your lab associated with gene expression, seed dormancy, and germination, which was very useful to the researchers like us in the seed-related aspect. Out in the concluding remarks. Of course, we will further look into this area and refer your research articles by you, ma'am. Thank you, ma'am, for sharing your valuable time with us. Department of Science and Technology, and all the participants. Gratitude to you, ma'am.
Thank you so much.
I would also express this opportunity to thank Dr. Rosemary Francis and Genetics Agricultural Research Station Manuti. Concluding remarks. And also, she is the working hand behind this entire webinar series and scheduled all this lecture series with eminent and experienced personalities in the field. Sincere gratitude to Dr. P.G. Bastin, Professor and Head, Department of Seed Science and Technology, who was given the welcome address, and Ms. Ajina Goban, former student of our department and now working in FCI, who is given nice introduction to our speaker, and all the staffs and students in the Department of Seed Science and Technology for their sincere efforts in successful completion of today's webinar. I also thank all the participants, including scientists, researchers, students, from in and outside the university for their wholehearted cooperation from beginning to the end of this webinar. Thank you. Thank you once again.
Thank you so much.
Thank you. We will meet very soon in the next edition of this webinar. Thank you all.
Thank you, Digi. Bye, Yasin. Bye.
Bye.
See you.